Cell culture is a cornerstone of modern biotechnology, driving the production of therapeutic proteins used in everything from vaccines to cancer treatments. Today, we'll dive into the role of cell culture in protein production, focusing on its mechanisms, applications, and ongoing challenges in the field. At its core, cell culture provides the platform for expressing recombinant proteins by introducing a target gene into mammalian cells, typically Chinese hamster ovary cells, or HEK293 cells. We can leverage the cell's machinery to produce complex proteins that require proper post-translational modifications. Moving beyond basic culture techniques, modern cell culture for protein production has evolved significantly. The use of bioreactors for large-scale, high-density cultures is now standard, but innovations such as single-use bioreactors and automated cell culture systems are pushing efficiency to new levels. In addition, fed batch cultures and continuous perfusion systems allow for more controlled and sustained protein expression, often leading to higher productivity compared to traditional batch processes. Despite these advances, a key challenge remains: maintaining cell viability and protein stability over time, as prolonged culture periods can lead to metabolic shifts and protein degradation. Once proteins are expressed in culture, the next critical step is purification. Techniques like affinity chromatography and ion exchange chromatography are routinely employed, but the complexity of the protein often requires additional steps, such as size exclusion chromatography. Additionally, quality control is essential, as even small variations in protein structure can impact its therapeutic efficacy. Methods such as mass spectrometry and capillary electrophoresis are used to verify protein identity, purity, and glycosylation patterns. The challenge remains to streamline these processes while ensuring batch-to-batch consistency in the final product. Cell culture's role in producing biologics has been transformative, particularly in the field of monoclonal antibodies and gene therapies. Monoclonal antibodies, such as rituximab and trastuzumab, are essential for treating cancers and autoimmune diseases, and their production relies on the precision of cell culture systems. Moreover, gene editing technologies like CRISPR are now being integrated into cell culture systems to improve protein production. By fine-tuning cellular pathways, scientists can create cell lines that produce therapeutic proteins more efficiently and with greater stability. Despite these advances, there are still significant challenges in cell-based protein production. The cost of production remains high, particularly in large-scale manufacturing. Optimizing cell line stability and productivity over time is still a major hurdle, especially when scaling from lab to industrial scale. However, the future is promising. As we continue to refine techniques like directed evolution, metabolic engineering, and high-throughput screening, we'll see improvements in both the efficiency and cost-effectiveness of protein production. Furthermore, new bioreactor designs and tighter regulatory controls will be crucial in meeting the increasing demand for biologics. Cell culture continues to be a driving force in the biopharmaceutical industry, enabling the production of therapeutic proteins with precision and scale. With ongoing technological advancements, the future of protein production is poised for even greater innovation. That's all for today's deep dive into how cell culture drives protein production. If you found this topic interesting and want to learn more about cell culture techniques and innovations, don't forget to like and follow. We'll be sharing more tips, insights, and knowledge on cell culture in future videos. Thanks for watching.